Good morning. Now it's working. Okay, great. Thank you. I'd like to welcome everyone to the session this morning, which is entitled GPCRs in Cancer, New Therapeutic Opportunities. My name is Paul Insel. I'm the sort of organizer of this, and there are going to be several speakers. I'm going to speak, and then there'll be several speakers after me. And the title of my talk, but it's also part of the introduction, is GPCRs in Cancer, Unknown Unknowns Waiting to be Discovered. And this particular session is sponsored by the British Journal of Pharmacology, to which we're very grateful for providing some travel money on behalf of the speakers, all of whom I think will enlighten you because of the different areas that each of them will cover. So in terms of the outline of this talk, I'm going to cover in the next 35 minutes sort of 5 topics. One, I'm going to mention very briefly GPCRs as drug targets. Then I'm going to talk about what we've coined as GPCR omics, and talk about the rationale, approaches, and major findings. And then talk about GPCRs with differential expression, in particular in solid cancer, solid tumors. And then briefly give 2 examples of work that's come out of our approach to using this GPCR omics approach. And in particular, I'll be talking about a proton sensing GPCR in pancreatic cancer-associated fibroblasts. And then I'll talk about a GPCR with approved drugs that might be repurposed to treat pancreatic cancer. Well, I think everybody who's interested in GPCR just knows that they are the most frequent targets of approved drugs. In this wheel diagram that's shown on this slide, you can see that they represent 35% of all approved drugs. And that's based on that proportion of both FDA and EMA approved drugs that target many major families. And that's because there are a large number of GPCRs, and they're very, very druggable, as everyone knows, and they tend to be safe. And also they work on the outside of cells, which makes them easier to get to targets. Now, what about new opportunities for GPCRs as drug targets? So in spite of the utility of GPCRs as drug targets, actually only about 20% of the 800 or so GPCRs have been successfully targeted. And this suggests the potential of perhaps many additional GPCRs. And so we've asked the question, might previously unrecognized, unstudied GPCRs be what's called unknown therapeutic targets? And some of you may remember the late U.S. Secretary of Defense, as well as other secretaries over time, Donald Rumsfeld, who during the Iraq War in the United States said that, as you can read with me, reports that say that something hasn't happened are always interesting to me, because as we know, there are known knowns. These are things we know we know. We also know there are known unknowns. That is to say, we know there are some things we do not know, but there are also the unknown unknowns, the ones we don't know. And that's what I would suggest are some of these GPCRs that we haven't really studied. So our lab has asked, might unknown GPCRs be new therapeutic targets? And what we've done is to have used unbiased approaches to assess endogenous GPCR in terms of their expression and action. We've termed this GPCR omics, and we've focused on native cell types. And we've used a variety over the last 10 years or so with data on various tissue fibroblasts, iPS-derived cardiac myocytes and cardiac tissue, adipocytes, neurons, and vascular smooth muscle. But today, I'm going to focus on cancer and cancer cells. And we've published several papers in the last few years related to that. In particular, the first example there is one in PLOS Biology, which very few people have read, unfortunately, even though probably because it's got about 100 figures in it, and it's got a lot of data. But the focus is mostly today on my talk will be on pancreatic cancer, and pancreatic cancer cells and cells in the tumor microenvironment, and in particular, cancer-associated fibroblasts. And what about this GPCR omics workflow? Well, basically, it's very simple. We're trying to find out what are the native expressed receptors. So we obtain cells or tissues from healthy or diseased subjects, cell lines. We isolate the RNA and generate cDNA, and then we perform RNA-seq and do a large number of reads, and then we break down and determine what are the receptors that are expressed. But most important, of course, is to validate these mRNAs through expression of protein, functional activities such as signaling and functional responses, and then going on to animal models and using human genetic analyses as well. Now, other GPCR-omic approaches exist, and they include the use of GPCR arrays, single-cell RNA-seq, which I think is not very beneficial, and I'll be happy to explain why in the question period, as well as proteomics, of which I'll allude to briefly later. So what are the general conclusions we've come to from defining these endogenous GPCRs in native cells? First, most cell types that we've studied actually express more than 100 non-chemosensory GPCRs. And many of them are these orphan GPCRs, whose physiological agonists we don't know. And data that we've followed up with GPCR arrays and RNA-seq data that correlate very closely along with independent qPCR analysis. The most highly expressed GPCRs endogenously are generally not ones that have been previously studied. And that represents, then, these unknown unknowns. Then the newly recognized GPCRs can alter signaling and cell function, and thus unbiased GPCR profiling, this GPCR omic approach, we think, can identify new to us as human beings and scientists, new GPCRs that regulate cell physiology. But really, the reason we got into this is we want to ask, if you compare healthy cells or healthy tissues with diseased cells or tissues, do we identify new GPCRs that can contribute to pathophysiology and be potential therapeutic targets? And as I've said, we focused on cancer. Now, what about GPCRs in cancer, which is the main theme of this session? And if you think about it, the growth and function of endocrine gland tumors, a number of them are regulated by hormone-activated GPCRs in the pituitary, adrenal, thyroid, ovary, testes. And GPCRs also regulate tumors that are of non-endocrine, pure endocrine tissues, endocrine hormone tissues, such as the prostate and the breast. Certain GPCRs and G proteins can be mutated in various cancers, and certain other GPCRs, such as chemokine receptors, which the next speaker later speaker, Martine, will mention, I believe, are known to regulate cells, especially immune cells, in tumors. But beyond endocrine and hormone responsive cancers, largely, GPCRs have been ignored in cancer therapy. And there's a number of reasons for that, which we can talk about in the discussion. But we've asked, could GPCRs in other cancers be therapeutic targets? And even in the absence of mutations, might GPCRs with increased expression in cancers compared to normal tissues influence cancer biology, and perhaps as functional oncogenes, if you will, on cancer cells, or on other cells in the tumor microenvironment, and thus be novel or new therapeutic targets for such cancers. So in preparing for this meeting today, in this presentation, I did a quick PubMed search to ask, I hit GPCRs in cancer, or GPCR in cancer. How many publications have there been in each of the last 7 years? And you can see that there's been a rather prominent increase here in the last 5 years. A little bit of a slip in 2022, maybe that's from the pandemic, but the numbers are over 200 GPCR publications related to GPCRs in cancer. Now, in further preparation for today's talk, being very au courant, I asked ChatGPT, please summarize the studies since 2018 regarding G protein coupled receptors in cancers. And I got a very interesting answer. I'm giving you the exact quote. Here is a summary of key findings from studies conducted on GPCRs in cancer since 2018. And each of these topics below, there were explanations given, which so made it pretty long. There's GPCR activation and cancer progression, GPCRs as therapeutic targets, GPCR-mediated immune response, GPCRs in drug resistance, GPCRs in cancer metabolism, and GPCRs in the tumor microenvironment. Again, actually capturing what GPCRs do to a certain extent in normal cells. But I wondered, and perhaps you're wondering, which specific GPCRs are they talking about? So that's part of what we've done. We've applied a GPCR omic approach using both bioinformatics and experimental studies. And the most major study we did was really done largely by a very talented postdoc whose picture is shown here, Krishna Sriram, in which he worked out a technique to examine 11,000 tumor samples from 45 different solid tumors for their expression of GPCRs, based on RNA-seq data that's in the public database, the Cancer Genome Atlas, and compare that to 9,000 control tissue samples from the GTEx database, and sort of essentially put those data together by analyzing a single optimized pipeline, and then began to ask the question. Mostly we focused on this lower right to ask the number of counts, the number of times the gene is detected, to look at differential expression between the normal tissue and the cancer tissue. And the answers were quite striking. And this is published in this PLOS Biology paper, which I've referenced here. In 45 types of solid tumors, many GPCRs, typically more than 35 per tumor type, showed increased expression compared to normal tissues, that is the GTEx database. And these, when I say increased expression, I mean more than twofold higher, expressed as at least one transcript per million, and a median expression, and a false discovery rate of less than 0.05. And you'll note this is just a partial list, and here's the most highest expressed ones, if I can get this to work, was testicular cancers, not surprising, I guess. But the other highest expressing a cell type, cancer type, was pancreatic ductal adenocarcinoma, about which I will say much more. So thus, it seems like there's many potential opportunities exist for such increased GPCRs, increase in expression, to be novel targets in solid tumors, or to be contributing to the biology of those tumors. So what does that mean, though? I'm just telling you in the tumor, and that could reflect the presence of those GPCRs in the cancer cells, but also in other cell types in the tumor microenvironment. And there are cell types such as stromal cells, which referring to as cancer-associated fibroblasts, vascular cells, immune, inflammatory cells. Many of them could express some of those GPCRs. And I'm going to tell you a little bit more about CAFs, because that's what I'm going to describe some data on in the next few minutes. And CAFs and their GPCRs are potential targets in fibrotic tumors, such as pancreatic ductal adenocarcinoma, breast cancer, and several others. And they can be an obstacle for chemotherapy and contribute to immune suppression, hypoxia, altered metabolism, and overall survival. And this is a CAF because the CAFs essentially can communicate through various mechanisms to all the other cells, many other cell types in the tumor microenvironment. CAFs themselves are derived from fibroblasts, and in some tissues from specialized cells known as stellate cells. So what about pancreatic cancer? I think most people nowadays know about it based on people in the popular press who have passed away with it, or members of their own family or friends. Certainly I have, and all of the above. And it's the most common type this PDAC, pancreatic ductal adenocarcinoma, is the most common type of pancreatic cancer. It's currently, in the United States, the third leading cause of cancer death, and it's predicted to be the second leading cause of cancer death during this decade. And the reason is because people are always often detected late, and there's been no major improvement in the 5-year survival. In the United States, the American Cancer Society got very excited because the 5-year survival went up from 11% to 12% last year. But that means still, 88% of the people are passing away. This is really a tragedy. And most people are diagnosed at an advanced age, so we need early diagnosis. And only about 15% are able to get this curative approach, would be surgical approach. What's shown in the diagram is that PDAC tumors classically sit in this little arm, if you will, of the duodenum in the gut, and they're in a retroperitoneal location behind. It's very hard for doctors or others to palpate them, to feel them. And they're often in what's called the head of the pancreas, perhaps not as common as in the tail. And so you can see that they can then immediately get up into the gallbladder, the bile duct, and immediately become metastatic within the liver, which those supply. A key feature of these tumors is fibrosis. It's been given the name desmoplasia in PDAC tumors, and current drugs have really limited impact on mortality, in part related to this desmoplasia. So clearly, finding new, effective, and safe therapies for PDAC is an important unmet medical need. So I'm going to tell you now, in the next phase of this talk, on example one, a proton-sensing GPCRs that's in pancreatic cancer-associated fibroblasts. And most of what I'm going to tell you is all done from human beings. And those involved in the British Journal of Pharmacology would be happy to know this is N of 5. That's sort of a small joke for those who understand that. But anyway, we did a study on primary cultures with my colleague Andy Lowy at San Diego, and found that 5 calf cultures were created. He created right out of the operating room, putting these cells into primary culture. And when they grow up a little bit, low passage, we looked at how many GPCRs they expressed. And each one expressed at about 100, of which 82 were shared in all of the various tumors that we saw in these patient-derived calves. And one particular receptor jumped out at us, especially when we compared its differential expression with the precursor cells, pancreatic stellate cells, so-called PSCs, or pancreatic fibroblasts that were not stellate cells. And that is this GPCR known as GPR68 or OGR1. And I must say, at the time when we saw this, I had no idea exactly what GPR68 is, and perhaps many of you don't know what it is. Well, GPR68 is a proton pH sensing GPCR. And it's a member of a GPCR family, which includes GPR4, GPR65, 68, and 132, that detects acidosis, that detects the low pH. And when you think about it, most tumors are in a low pH environment, which immediately sort of made some sense that nature would potentially use a tumor, create a response to the environment in which it lives, which is in a low pH environment, typically less than pH 7. As shown in this diagram, among the 4 GPCRs that I mentioned, only GPR68 was elevated, among the other, not the other 3. So it might be that PDAC tumors have evolved to sort of use their low pH via this calf-expressed GPR68 to contribute to tumor biology. Is that what's going on? Well, this very busy slide, which I sort of apologize for, is an alternative way to do single-cell analysis. It's a deconvolution of RNA-seq data in which one looks at what's the association between very good markers for particular cell types, cancer-associated fibroblasts, tumor epithelial cells, immune cells, endothelial cells, et cetera, and to say which GPCRs are associated with those cells. You might say it's a poor man's single cell analysis, but it's actually, some people feel, a better way of analyzing for entities like GPCRs that are not highly expressed. And what do we find? Well, in the cancer-associated fibroblasts, GPR68 is right up there, right there. But interestingly, GPR65, its cousin, is also highly expressed preferentially expressed in the immune cell population. And one could look at other cell types as well. Now, this is all data that's kind of bioinformatic-y, but what about looking at real data from patients? Well, a former postdoc in my lab in Japan, who's a pathologist, looked at a series of patients with pancreatic cancer and found, I think if you focus on the right side, a rather co-expression, if you will, of GPR68 along with alpha-smooth muscle actin, a fibroblast marker. And interestingly, he found that in 32 out of 38 samples, and all the other 6 looked different as a kind of cancer. They were not cells that had very much fibrosis. And we've conducted, and I'm not going to spend the time on this, not only because it's published, but I think it's the conclusions what's more important. We did in vitro studies with these patient-derived pancreatic stellate cells or patient-derived pancreatic casts, and we did gain-of-function studies, and we found that transfecting GPR68 into stellate cells increased the expression of IL-6, interleukin-6, and a whole series of fibrotic markers that are shown on this slide. And the converse was also the case. If we took calves and we knocked down in loss-of-function studies with siRNA the expression of GPR68, we blunted the expression of everything that I've shown above, multiple profibrotic markers and IL-6 as an immune product. And if we knock down GPR68 in calves that were growing in culture, and took the conditioned media out of them, we found that conditioned media could normally promote PDAC cell proliferation. But if it was done in cells that had a knockdown GPR68, there was much less proliferation. And so I'm not going to have time to go through this. I'm going kind of quickly, but to give you just the feeling of this, it would appear that there is a bilateral communication between PDAC cells through, in fact, TNF-alpha to induce the expression of GPR68 through NF-kappa B. And now, in turn, when GPR68 is in the membrane, responds to low pH, it couples to GS to erase cyclic AMP and pKa and Krebs, and it gets an induction of IL-6, which then comes out and is able to stimulate the growth of PDAC cells. And so data in support of this is in the paper I mentioned and also in a US patent that we just recently received. So that's all in vitro data. What about in vivo data? This is all completely unpublished, only been shown once in public before. What we did is we used a mouse that's called the KPC mouse, that is a genetically engineered mouse that has a mutation of KRAS, which many patients have as well. It has a knockout of P53, and it has an agent that allows the receptor the expression of this to be limited to the pancreas. And these tumors, these cells, these mice rather, spontaneously develop PDAC tumors, and we bred them to mice that have a knockout of GPR68 in the pancreas. And what do we see? Well, at 9 weeks old, these animals have a very large tumor, and we found that the tumor weight overall is 40% less if the animals have been bred to with GPR68 knocked out. You can see this is data for males and females, 22 controls and 22 treated with knockout and 41 controls. And this is for males and females, and you can see males and females both show a very similar response. So what are we doing about this? Well, in order to go further to develop a drug, potentially, to begin, because we think GPR68 is an interesting target, we've developed a partnership between my lab, University of California, and a group called Audubon Labs, about a year and a half ago. And they don't believe anything we all do, as you may know. And so they've repeated all the experiments that we did, plus some, and they confirmed everything that I just told you very quickly. And they've extended the data on that, and they found that GPR68 is expressed in calves from multiple types of solid tumors. There's a low pH activation of signaling and a series of responses in GPR68 expressing cells. And the high throughput screen is in progress to identify drugs that may be able to blunt the activity of GPR68. If you're interested, Audubon is actually has a venture capital. It's sort of a group that tries to work with early academic investigators. And if you're interested, I'm happy to talk to you about them afterward. Okay, well, what about GPCRs in pancreatic cancer cells? I've just talked about in calves. What about in cancer cells? And the second example I'm going to tell you in the few minutes I have left is a GPCR with approved drugs that might be repurposed, we think, to treat pancreatic cancer. And believe it or not, we focused on the histamine receptor, HRH1, which is a GPCR, which I'm sure some of you take antihistamines, but for HRH1 for allergies. And yet, we think that this could be a target for pancreatic cancer. Why do we think so? Well, here's the data from the TCGA data, Cancer Genome Atlas. This is the data from 147 patients, expression. This is the data in normal people. There's hardly any overlap in any of them, 165 samples, and it's around 20-fold higher expression in the samples from the patients. And if people who have less expression live longer than those who have higher expression. And I might point out that in more bioinformatic analysis, the data indicates that this HRH1 is preferentially expressed in the pancreatic cancer cells and not other cell types. But there's other data in the literature, which I'm just quoting from, that sort of supports this idea. The progression of PDAC tumors correlates with the infiltration of mast cells, which some of you may know are the cells that both make, store, and release histamine. And that's been reported a few years ago. And other studies in people have shown that those who are over 70 years old, especially those of us who are over 70 years old, who have allergies and use HRH1 antihistamines have a lower incidence of pancreatic cancer, as published a couple of years ago. So it turns out, when you look at human PDAC cell lines and organoids, they all have a high expression, shown here in a gene expression shown down below here. This is all HRH1, all these green things, and the other lower than expression are the other three histamine receptors, HRH2, 3, and 4. Similarly, we can find in various cancer cell lines, human cancer cell lines, decent expression, high expression of these particular receptors. And more importantly, in work with Hervé Thiriac, he has found, in making organoids, that when you take tumor organoids versus normal organoids, there's a prominent increase in expression of HRH1. But is it functional? Well, here's something that I thought would be appropriate for this Congress. This is the ability of histamine in there's three different cell lines to increase calcium in a FlexStation assay, and the ability of fexofenadine, which some of you may know as Allegra, to block the activity to block the increase in calcium. And the values for the EC50s and the PKIs are very similar to data that's in the Guide to Pharmacology, which we wanted to mention. And in addition, data I'm not showing you is that the inhibitor YM254890, which is known to be an inhibitor of GQ-G11, completely blocks out the ability to increase in calcium. We also have done experiments which don't come out quite as well as I'd like, looking at scratch assays, and this is histamine's ability to show that the cells migrate in culture, and they can be blocked by, in this case, a clemastine, and in this case over here, fexofenadine, very nicely, where they don't have much of an effect. So the cells show migration in response to histamine activation. And in studies that we've conducted with Elena Thomas-Bord and Richard Gross and Peter McCormick at Queen Mary, they also have a very nice setup to analyze invasion. And you can see histamine in PANC1 cells and ASP1 cells, other cells, have a prominent increase in invasion, which is totally blocked by fexofenadine. And not shown here, we can also induce this with a 2-PEA, which is a HRH1 selective agonist. But we've gone on to do studies in these KPC mice. And amazingly, they have extremely high expression of HRH1, relatively low expression of the other, there's HRH1, a low expression of the other types of histamine receptors. And much like I just showed you, they have the ability to increase calcium, and that's blocked out by in this case, loratadine and clemastine. Now, also, it turns out that we looked at the expression of mast cells in the tumors, and we find, as shown on the right side of this slide, by staining with avidin, avidin-rotamine, which essentially stains for mast cells, much higher content of mast cells in these cultures, or in these tumors, excuse me. And so we've actually done a modest amount of in vivo data so far. We found a technique where we could put fexofenadine, Allegra, in the water supply of mice and let them just consume that and see what the impact would be. And we gave that to these KPC mice. Or we gave them the solvent DMSO at low concentration, the one used for fexofenadine. It didn't change their drinking. It didn't change their ability to function in any other way. And we thought it was very important to document that we actually raised the blood level and raised the level within the tumors itself. And interestingly, statistically significant, there is around a 20% decrease in the pancreas weight, the tumor weight, in animals that have received fexofenadine. So this is sort of a treatment that we begin at around 4 weeks and follow them over 5 weeks. In addition, we've taken those tumors out and we've looked at gene expression, and there's a decrease in PD-1, in VISTA, which has been described by others, being regulated by histamine, collagen 1A1, and IL-6. So we think, we're not sure, we think that there may be a mast cell histamine HRH1 axis in these pancreatic cancer cells. And it implies the potential that one might be able to use HRH1 antihistamines to treat PDAC, and that's what's shown in this diagram. Mast cells making histamine, activating the receptor that activates phospholipase C and increases calcium, and that activates various transcription factors to influence cytokines, which I haven't shown you, migration, invasion, and this can be blocked by, we think, by antihistamines. So in summary, what I've tried to tell you is that unbiased identification, quantification, what we call GPCR omics, of endogenously expressed GPCRs reveal new GPCRs that regulate cells in health and disease, including in cancers, that non-mutated GPCRs with increased expression can be functional or potential therapeutic targets for cancers. GPR68, a proton-sensing GPCR, differentially expressed and functional in these human calves, utilize low pH in their tumor microenvironment to regulate the calves and tumor growth, and it's thus we think is a novel therapeutic target for PDAC. Collaborative work with Audubon Labs confirms, and that is my conflict because I'm involved with them, I should have said that earlier, confirms findings reported here and seeks to identify compounds that will blunt the low pH GPR68 GS cyclic AMP mediated responses in PDAC and perhaps other cancers. And lastly, that HRH1 is a highly expressed and functional in PDAC cells and tumors and implies that HRH1 antihistamines might be repurposed for the treatment of PDAC patients. So the general conclusion that I take-home message is that certain GPCRs are differentially expressed and functional in cancer. I've only shown you data for a couple of them. Such GPCRs are examples of these unknown unknowns. Didn't know it unless we looked for it, but had to use an unbiased way to find it, where further study has the potential to yield new therapeutics, likely as a part of combination therapies, not likely to work alone, but as combinations to treat cancers. And lastly, let's come back to ChatGPT. If asked, remember, summarize the results since 2018 regarding G protein-coupled receptors in cancers. I didn't tell you the last part of what it said. Overall, research since 2018 has expanded understanding of the roles of GPCRs in cancer. These studies have identified several GPCRs as potential therapeutic targets and highlighted their involvement in various aspects of cancer biology. And this last sentence, this is ChatGPT, it's not me. Further investigation into GPCRs is expected to provide valuable insights for the development of novel cancer therapies. And lastly, I want to acknowledge again BJP for providing support for this, and the people in my lab, in particular Sue Wiley and Christina Samaran and Krishna Sriram, some I mentioned, who've pioneered in these studies that I've showed you, along with others from my lab and collaborators, including Andy Lowy in his lab, and Hervé Thiriac and John Weitz. Anna Donardo helped with the mast cell analysis, and Jin Zhang and Clara Posner and Eric Greenwald, some studies of some cells that express high expressive of GPR68. And I mentioned Hiroshi Nishihara, as well as Peter McCormick and Richard Gross and Elena Bird. Thank you very much for your attention, and I'm more than happy to answer questions.
Before we ask questions, Paul, thank you very much for this amazing lecture. And I must say, so I didn't have time to introduce him, but so he's a distinguished professor at UCSD, being trained first at the University of Michigan through clinical training, then moved to the NIH, UCSF, and then UCSD. And I must say, he has been a driver of this field. He put forward the GPCRs in cancer, and I'm glad that also ChatGPT is agreeing with that. So please, questions.
I think, can you get a mic? They want to use a microphone.
Sorry, can you use the microphone so everyone can hear?
So let's first go to microphone 2.
Oh, sorry. Yep, okay. Hi, Paul. Thank you very much for that. Nicola Smith from Sydney.
Hi.
I should ask GPT, but I'm going to ask you instead. I want to know about the known knowns. So my graduate student, Mariah Stavry's got a paper today where we were interrogating the RNA-seq databases because everyone says sweet taste receptors are important for cardiometabolic diseases, but we can't find it. Have you ever, in the human genome, in those tissues, so have you ever come across the problem with the known knowns where people say it's there and you've gone through your data sets and you haven't been able to validate the expression?
I couldn't quite hear the 9 nines?
No, so that's my Aussie accent. Oh. Donald Rumsfeld, the known knowns.
Oh, the known knowns.
Yeah. So where it's just assumed, and have you found that things that didn't appear, you've actually been able to validate experimentally but weren't in those databases?
Well, Not really, that I can remember. Pretty much everything that we've followed up on does. Now, someone is always thinking, well, wait a minute, you use mostly RNA, what about the protein?
Yeah.
We did have one example of something that looked like it was very high in the RNA, but we didn't see the protein, and so we dropped it. But other than that, that's the only example that I can recall, Nicola.
Thanks, Paul.
Rachel, you had a question?
Is GPR68 is a orphan receptor? GPR68?
Well, it was originally described quite a few years ago, and the activation by low pH has been confirmed by lots of people. There was one study that suggested there may be some peptides that can activate it, but there's some question about some of those data.
What is a known ligand of GPR?
No, the low pH, hydrogen
pH?
ions. Hydrogen
High
ions.
pH?
You know, low pH, hydrogen. So if you take the cells and you move them from pH 7.5 or 7.4 to 6.5, the calcium, as I showed you, I didn't show you, but calcium can shoot up, but cyclic AMP goes way up, and it's low pH that's stimulating.
So in view of the time, let's go to the other people. Just Jill.
Jill? I can't hear you. Is your microphone on?
I'm a bit short. Is that better?
That's better.
So a philosophical question, if I may. There was a previous symposium at this conference which looked at beta blockers in cancer. So beta blockers are well tolerated and are very cheap. Antihistamines are well tolerated and very cheap. Pancreatic cancer is deadly. Chemotherapy costs a lot in money and a lot in side effects for the patient. Is there an argument that when you get a diagnosis of a cancer, you should have an antihistamine and a beta blocker from the outset?
Yes, there isn't. I mean, I've actually hinted to people they might want to do that. Tim Wang, a guy from Cornell, who gave a talk at our school a few months ago, he described some experiments doing beta blockers and getting a very nice effect in animals, and looking at patients who'd used beta blockers, getting an effect. And I said to him, you know, don't you think we have to do a clinical trial? And he says, nobody wants to support that because they're not going to make any money. So that's the problem. So how do we actually do that? One thing that we need to do, clearly, is to now do more studies with the standard of care, gemcitabine, and show that that is additive or adds to that. But our animal data for both of the examples I showed you looks pretty good. So, and histamine looks pretty good. It's doing some things that are important. So I share your cynicism, and I worry that people won't do it unless some of us can figure out a way to get the money to do it.
Absolutely.
So short question for microphone number 4.
Karen Gregory, Monash University. Thanks, Paul. I was wondering if you've looked at if there's increases in the ligands, so the enzymes that create GPCR ligands or the peptides themselves.
No, that's an excellent question, and thank you for asking that. Especially the ligand, in this case, histamine. We looked at it, and it is not it's not high in the animals. It's not very high, but we don't think that that you can get an effect unless you add the histamine. In other words, that there's not an automatic synthesis by the cells of this. And as I said, it's the mast cells that create the histamine, not the cancer cells themselves. But we have looked at that.
Last question, microphone number 3.
Thank you. Michelle Halls, Monash. Just to follow on from Karen's question, do you see changes in the signaling pathways downstream from these receptors so you get amplification at that level, or is it all driven by the change in expression?
I'm not sure, Michelle, I get your question, but I can tell you that, like, is it all turned on? Like, for example, if 68 works through GS, is GS generally activated?
Yeah, so
No.
is there dysregulation of the, so maybe you get more cyclic AMP than you normally would?
It doesn't appear to.
Okay.
But we haven't maybe done the exact way what you're asking, but it doesn't look that way.
Thank you.
Okay, once again, thank you very much.
Thank you. Thank you. So I'd like to invite Rachel. So this is Rachel Bar-Shavit from Israel. We are joined now by, and she studied at the Weizmann Institute and completed her PhD studies and postdoctoral fellowship at Washington University, and has been a leader in looking at particular GPCRs, which I think she'll tell us about, are the protease-activated receptors. So thank you, Rachel, for doing it.
Good morning, and thank you, Paul, for the invitation to participate in this morning session on GPCRs in cancer. As an attribute to the canonical presentation by Douglas Hanahan and Bob Weinberg, who presented the various schemes in cancer, Silvio Gutkind has shown the involvement of GPCRs in different cancer phases, including F2R, PAR1. I will focus in my talk on protease-activated receptors, PARs, and tell you about the endeavor to allocate PAR members as cancer driver genes. Looking at Kaplan-Meier's plot, in this case for breast cancer, for example, it is shown that whenever PARs are overexpressed, there is worse survival time. Using another web server of gene expression profiling interacting analysis, it shows that PAR members are highly expressed in serous cis adenocarcinoma of ovarian cancer, most notably PAR2, which is also recapitulated in the worst survival time observed in breast cancer for PAR2 when it is highly expressed. And indeed, we and others have demonstrated that PAR2 is a dominant among the PAR members. Jitendra Nag, a postdoc in my lab, have demonstrated that in fact PAR2 is a stem cell cancer marker capable of self-renewal and tumor growth. This is shown in an animal model whereby using limited dilution assay, in this assay, one can analyze the minimal number of cells that are still capable of self-renewal and tumor generation. As can be seen here, HCT cells that highly express PAR2, as minimal as 10 cells can generate tumors, while where knockdown of PAR2 shows a requirement of 1,000 cells in order to produce a tumor. And indeed, when we have analyzed series of cancer stem cells, it is shown that when we knock down PAR2, several stem cell markers like LGR5, CD44, CD133, CD166, ALDH1 are downregulated, are inhibited when PAR2 is knocked out. The high invasive metastatic potential of PAR2 is demonstrated best in a spleen-liver model, whereby one inoculate tumor cells to the spleen and look for metastasis in the liver. We have demonstrated that by inoculation of cells that express high level of PAR2, to the spleen, one can see multiple metastases to the liver as compared to knockdown PAR2, where you can barely see primary tumors, nearly none, and very few to none metastases to the liver. In another mouse model of wild-type mice and knockout PAR2 mice, when we ectopically inserted the colon cancer, foci in the colon. One can see following AOM DSS model by injection of AOM followed by 3 sessions of DSS. One can see multiple colon foci, colon cancer foci. In comparison, in the PAR2 knockout mice, nearly no, very little of colon cancer foci are seen. So overall, in this model, we demonstrate that PAR family and PAR2 as an exhibitor of the PAR family, representative of the PAR family, play a central role in tumor biology. And from this, Miriam Maoz and other lab members have studied the molecular mechanism of PAR-based mechanism, and we have identified plextrin homology pH binding motifs within the C-tails of PARs. These motifs enable signal proteins that contain pH domain, plextrin homology domain, to associate directly with the PARs. We have fished out by protein-protein antibody arrays several signal proteins of pH domain, and we have identified the minimal binding region in the C-tails of PARs, which associate directly with a panel of signal proteins containing pH domain, such as AKT, ETK-BMX, SOS1, etc., also GAB1. And when, for example, we have inserted mutations to the pH binding domain via sequential AA residues, we can demonstrate that while the wild type of PAR1 in this case can associate with ETK-BMX via its pH domain, the mutant form can no longer bind and form this complex. Here shown controls for equal expression of the mutant and the wild type. And when we have looked at the mouse model following orthotopic injection to the mouse mammary gland, we see large tumors in the presence of PAR1 and ETK-BMX as compared to very small little tumors in the mutant form. This is comparable to empty vector-injected mice. So this was shown for PAR1, and the same is shown for PAR2. When we inserted mutation in the pH binding domain of PAR2, we see smaller tumors as compared to another tumor, another mutant, which does not have an effect on tumor generation, as shown here, large tumor formation and very small tumors in the presence of H349A mutant, comparable to a truncated form of PAR2, which is incapable of inducing signaling. Here shown tumors generation of wild-type PAR2 versus the mutant H349, incapable of associating with signal proteins. So from this, we have selected a family, together with Chaim Gilon and Amnon Hoffman, we have selected a library of cyclic peptide directed to the pH binding domain. And when we have analyzed the linear peptide, the basis of this cyclic peptide, we see that the chemically modified linear peptide can well penetrate to the cells and efficiently abrogates the association with ETK-BMX signal protein, and also inhibits efficiently matrigel invasion assay, as shown here, matrigel invasion via activation of PAR2, whereas it is completely abrogated in the presence of the linear peptide based on PAR pH binding domain. So we have selected a lead cyclic peptide named with a ring size of 4 by 4, named PC4 x 4. And when we have analyzed this cyclic peptide in mice, we have started injecting the treatment with the cyclic peptide only when we saw a large tumor formation. And then we have started the treatment and see very efficient tumor reduction following the treatment with the PC 4 by 4. It is well known for a while that GPCRs can crosstalk with EGFR receptor, EGFR and RB family. And this is shown also, known true also for PARs. PAR, via direct association with SOS1 and GRAB1, can directly tyrosine phosphorylate the EGF receptor. And indeed, merely by activating PAR4 in this case, with the activating ligand, we see potent induction of the tyrosine phosphorylation of EGFR. And this is completely inhibited when we introduce mutation to PAR4 pH binding domain. And indeed, following treatment with the cyclic compound, we see potent inhibition as compared to tyrosine phosphorylation of EGFR. This opens a wider horizon for the treatment with a PAR-based therapy, also for EGF receptor expressing tumors, RB family EGF receptor. And indeed, we demonstrate here that high level of PAR, in this case PAR4, high level of PAR4 is expressed in tumor tissues of HER2-new breast cancer, as also triple-negative breast cancer, all expressing high level of PARs. Another angle of our studies is looking at developmental stages, which are very nice to look at along with tumor progression events. One can look at mammary gland of a mouse, developmental stages, which all takes place after its birth. One can see the alveoli, ductal side branching, milk production proteins, which goes back following apoptosis to alveolar buds. So these cycles take place in a mouse after its birth. And this is a very efficient model to look at tumor progression as well. So we have generated transgenic mice overexpressing PAR1 in the mammary gland. And when we looked at the protein profiles, we observed a remarkably high level of beta-catenin in the transgenic mice versus wild-type mice tissue. This is true for 8 weeks puberty, 10 weeks, 13 weeks, pregnancy 4 days, and pregnancy 8 days, all show remarkably high level of beta-catenin as compared to wild-type tissues. So this led us to study PAR-induced beta-catenin stabilization pathway, which merely by activating PAR2, it leads to beta-catenin stabilization, which enters the nuclei and becomes a transcription factor responsible for an oncogenic gene signature downstream. This is similar to Wnt-Frizzled association. Frizzled, another 7 transmembrane domain receptor activated by Wnt ligands, which leads to beta-catenin stabilization and beta-catenin transcriptional activity, which is a major pathway in tumor biology. Jitendra Nag have shown that another layer of regulation of PAR takes place. And indeed, he demonstrated that RNF43 and E3 ubiquitin ligase is a negative regulator of PAR2. This was studied from an RNA-seq analysis comparing between high-level PAR2 cells as compared to PAR2 knockdown cells, showing several interesting genes, but most noticeably, RNF43 shows inverse correlation with PAR2 expression. When PAR2 is knocked down, RNF43 is overexpressed. Indeed, we could demonstrate, this was verified by real-time PCR analysis, and we could demonstrate that biotin-labeled cell surface PAR2 is degraded in the presence of RNF43. I'm not doing well with the mouse here. Anyway, this is also, this was confirmed by FACS analysis and also showing the ubiquitination of PAR2 via RNF43. What are the functional consequences of this negative regulation? It shows that while PAR activation leads to beta-catenin stabilization, in the presence of RNF43, it is completely inhibited, and also the transcriptional activity of beta-catenin shown by left TCF top flash assay, while it is upregulated following PAR2 activation, it is completely inhibited in the presence of RNF43. And what are the implications of this RNF43 regulation? It shows that PAR2 is in fact another target for RNF43 degradation. And it shows here that it competes with Frizzlets on RNF43 pool. While PAR2 levels are nearly 42%, in the presence of RNF43, it is reduces to 22%. However, in the presence of Frizzlets, it increases back to 36%. So, PAR2 RNF complex are similar with Frizzled RNF43 complex, competing for the same pool of RNF43. What happens when RNF43 is mutated, as we see in several cancers? Upon mutations of RNF43, both levels of PAR2 and Frizzlets are increased. And indeed, RNF43 acts as a tumor suppressor gene. It is highly expressed in cases of very small tumors or normal tissues, but it is not expressed in highly metastatic tumors. And also, this is in the spleen liver model, also in another model of the wild type and PAR2 knockout mice, it is highly expressed in the knockout mice, showing very little small tumors, but it is not expressed in the, or very low expression is seen in the wild type metastatic, in the wild type colon cancer foci. So what is our current understanding on PAR regulation in the context of cancer? PAR2 is expressed on the cell surface, and in the presence of RNF43, it is ubiquitinated and degraded via the proteasomal system. However, it can be rescued in the presence of LGR5, another 7-transmembrane domain receptor, and the stem cell marker, which is, when ligated to its ligand R-spondin, RSPO, LGR5 sends RNF43 for ubiquitination and degradation, and therefore, PAR2 remains high and increased level, as also signaling of beta-catenin and beta-catenin transcriptional activity. So what are the current facts that we know? Inactivating endogenous mutations of RNF43 are found in cancers of colon, pancreas, endometrium, and ovaries. What can we learn from these RNF43 mutations in cancer? In the presence of mutant RNF43, the levels of PAR and Frislets are high, as also beta-catenin signaling. This mutation can, in fact, identify a patient population that will best respond to both PAR-based therapy as also upstream Frislet inhibitors. So our conclusion and future direction, in the combat of cancer, it is yet challenging to define an appropriate patient population for the development of suitable therapeutic medicament. As well as others, have demonstrated the central role of PARs in cancer as recapitulated by PAR2 that functions as a cancer stem cell gene capable of self-renewal and tumor growth. We have elucidated pH binding motifs within PAR C-tails as a platform for drug design. Selection of a lead cyclic PAR-based compound for therapy in tumors expressing PARs as also EGFR RB genes. Lessons from transgenic mouse revealed that PARs induce beta-catenin stabilization, a central pathway in tumor biology. RNF43, a ubiquitin ligase, is a negative regulator of PAR2, similar to its regulation on Frislet Wnt-induced beta-catenin pathway. Endogenous mutations in RNF43 are found in tumors of the gastrointestinal system, endometrium, and ovarian cancer. It allocates PAR-based therapy as an important therapy target for patients with RNF43 mutation. Thank you. I would like to thank our lab members here that participated in the study.
So this paper is now open for discussion.
Thank you for an interesting speech. My question is related to physiological activation of PAR receptors caused by proteinases like thrombin, tryptase, etc. So, can't you hear me?
Yeah, I hear you.
Oh, good. But do you think that there is an uncontrolled overexpression or generation of some proteinases, or will overexpression of PARs cause a proteinase-independent PAR receptor signaling?
Well, you know, I've been asked exhaustively, excuse me, about thrombin function in this system. I mean, we don't see any effect on thrombin. Thrombin does not have, I mean, in the mouse models, no clotting activities. We don't see any of this. Now, PARs are overexpressed in epithelial malignancies. Normal epithelial cells don't express PAR. We see high expression level of PARs only in epithelial malignancies, a wide range of breast cancer, colon cancer, ovarian cancer, etc. All the epithelial malignancies overexpress PAR family, especially PAR1, PAR2, and PAR4. This is what we see.
So it may really be an proteinase-independent PAR receptor signaling. That's interesting.
It is possible that proteinases activate some other cell surface receptors, but we see abundant immobilization of proteases to the microenvironment, and these immobilized proteases well activate PARs. Not only thrombin, but many other, via biased activation, many other proteases activate the PAR family.
Okay, thank you.
Thank you for your talk. I was really interested in the signaling proteins that you have identified that interact with the C-terminal.
Excuse me, I can't hear you.
Sorry. I was interested in the signaling proteins that you found that interacted with the C-terminal tails of the PAR receptors. I was wondering if they interact with the receptors at rest or if it requires receptor activation.
I'm not sure I understand what you ask. How do we see the signaling of PAR?
No, sorry. The proteins that were interacting, was it by the pH domains, AKT and GAP1?
Yes, GAP1.
Yeah. So do they interact with the receptors without receptor stimulation, or do you need the receptors to be activated to see?
We see following activation,
Right, okay.
association of GAP1
Yeah.
with PARs. We
Okay.
see a complex via immunoprecipitation, via colocalization, immunofluorescence. Many aspects you can find the complex. And this takes place only after activation of PARs.
Thank you.
Can I just ask you a really quick question?
Yeah.
It relates to what somebody asked earlier. So what about PAR-1? I mean, PAR-1, unlike PAR-2, has an approved drug, Vorapaxar. Have you ever thought of, is that something that you could actually test if it influences any of the cancer models that you've looked at? Vorapaxar?
PAR1?
Yeah, and you focused on PAR-2, but I'm wondering about PAR-1.
Well, you know, we have started with PAR1 as a representative of the family, but it turns out that we came to learn that PAR2 is a dominant member of, it affects PAR1. I mean, we need the presence of PAR2 signaling in order to get PAR1 function. So transgenic mouse were made for PAR1, but then we started focusing on PAR2 because this is a dominant member. It turns out, not only our studies, but others. Also, Koleopoulos have shown that PAR2 is dominant over PAR1 in mouse intima blood vessels. So this is a concept now, that PAR2 is a dominant member among PAR family.
Great. Thank you very much, Rachel.
Thank you.
Thank you. The next speaker today is Ameera Jailani, who's submitted an abstract, and this is selected from the abstracts that were presented for the meeting, and so this is a somewhat shorter talk. And the title of her talk, as you can see, is The Effect of Adrenomedullin and Its Receptors in Pancreatic Cancer Progression and Chemoresistance.
Hi, thank you. So, yeah, I'm going to try not to repeat a lot of the things that Paul had talked about during his talk. So, first slide. Every year, there are almost 500,000 cases of pancreatic cancer worldwide, and almost the same number of deaths as well. So, almost everyone who gets diagnosed with pancreatic cancer would probably eventually die from it. Incidence rates has also been increasing by 10% over the past decade. So this is due to the fact that there's increase in risk factors, so such as obesity, as well as the fact that people are dying less from other cancers as well. Oh God, I just ended my slides. Okay, so if you look at this graph here on the right, you can see that whilst the survival rates for other cancers have improved a lot since the 1970s, survival for pancreatic cancer actually hasn't improved that much at all. And this is because that most of the people are diagnosed at late stages, and surgery is just not an option for these patients. So a lot of the treatments available for pancreatic cancer are palliative rather than curative. So it's currently classed as an unmet clinical need, and we need to find new ways to target this disease. So literature has shown that this hormone called adrenomedullin, or AM, is upregulated in many cancers, including pancreatic cancer. So normally what AM does in the body is that it's a vasodilator, so it's very important for vascular homeostasis. And this can affect well, this regulates our blood pressure, hormones, as well as placentation. But when AM signaling is dysregulated, this can also promote a lot of the different signaling that happens within the tumor microenvironment. So AM is important both for physiology as well as pathophysiology. So if we were to block AM as a whole in cancer, this could lead to adverse effects in cardiovascular as well as endocrine functions. So thankfully, AM has 2 receptors, and if we were to limit the block only to the cancer signaling pathways, we should then just target the specific receptor that mediates these processes. Unfortunately, AM receptors are slightly complex, so it's made up of 2 components. One component is a class B GPCR called CLR, or calcitonin receptor-like receptor, and the second component is an accessory protein called RAMPs, or receptor activity modifying proteins. So CLR on its own is not actually a functional receptor. It needs to partner with one of these 3 RAMPs in order to become a functional receptor. And each of these 3 receptor complexes you see here, they're all very, very different in terms of pharmacology. So they bind to ligands with different selectivity, and they also mediate a lot of different downstream signaling consequences. So in 2015, our group was awarded £5 million by Wellcome Trust to develop a drug for pancreatic cancer. So we hypothesized that if we were to target just the cancer promoting receptor, which in this case is the AM2 receptor, but if we were to leave the other one alone, which is the AM1 receptor, important for our normal functions, then this would present as a novel therapeutic strategy. So the development of this small molecule antagonist against this receptor complex is quite a long story, but we basically started from a known chemical entity, which are clinically approved CGRP receptor antagonists. So these are approved to be used to treat migraines. So what we did was we used a rational-based drug design to tweak the compound, and eventually we ended up with something that is selective for the AM2 receptor, with 1,000-fold selectivity over the AM1 receptor, as you can see from this, from the PIC50 values here. So these are the first-in-class AM2 receptor antagonists. So it's not just us. Independent groups have also shown the role of AM as well as its receptor components in cancer, pancreatic cancer specifically. So this first paper here shows that in patients with pancreatic cancer, the serum levels of AM are much higher than normal patients. There are also a lot of other papers that have tried to give insight on how AM can specifically mediate some of the pro-tumoral consequences. But it's this paper here by Dai et al. That is actually the first one to show the role of AM2 receptor antagonists specifically. And this came out well after we started the program. So it's nice to see that it supports our working hypothesis to target AM2 receptor in pancreatic cancer. So we got a compound that can antagonize AM2 receptor. So we want to validate its anti-cancer effects. So one of the first things we did was to get a panel of pancreatic cancer cell lines. So all of these represent the main genetic complexities or mutations within pancreatic cancer. So we did endpoint PCR, Western blots to see if they express AM or the individual receptor components. And you can see that they do express them, albeit to different extents. However, we don't know if they express the functional version of these receptors where the CLR and the RAMP are actually in complex with each other. So we next did a second messenger assay. So we did a cyclic AMP assay using the same panel of pancreatic cancer cell lines. So what we did was to use the family of CLR ligands this time, including AM, and to see whether they can induce cyclic AMP production in the cells. So you can see that even though all of them express the individual receptor components, they are actually differentially stimulated in terms of producing cyclic AMP. So admittedly, cyclic AMP, whilst it's the most established second messenger pathway for these family of receptors, it's not the only one. There's a lot more complex signaling happening, including other second messenger pathways like AKT, ERK. So we are also looking into that further. So here you can see some of the anti-tumor effects of the AM2 receptor antagonist, where in vitro it's shown to decrease the viability as well as increase the apoptosis of pancreatic cancer cell line. And these results have translated in vivo as well in a pancreatic cancer xenograft model, where the tumor burden is less in the mice that were given the AM2 receptor antagonists, and they survive for longer as well to a humane endpoint. So we did some histology, and it turns out that the effect that we see is not only on the cancer cells themselves within the tumor microenvironment, where we see decrease in proliferation, increase in necrosis in the in vivo tumors, but there is also an effect on the stromal cells, so the non-cancer cells in the microenvironment, where you can see here that blood vasculature as well as cancer-associated fibroblasts or CAFs expression has also been decreased. And this is quite exciting because, as Paul mentioned earlier, pancreatic tumors are very fibrous. Like 80 to 90% of the tumor is actually fibrous tissue. So that's one of the reasons why it's so treatment resistant. And this data as a whole suggests that we could use AM2 receptor antagonists in combination with some other kind of therapy, like chemotherapy, to allow them to actually penetrate into the tumor to exert that intended anti-tumor effect. And this effect that we saw on the CAFs was, it also was supported by the data in the Dye paper, where they found out that when they inhibited the AM-REM3 system, they saw less CAFs in metastatic sites, and this resulted in less metastasis of pancreatic cancer to the liver. So we decided to follow up with these observations. So I'm going to show you some unpublished preliminary data. So we also tried to see if fibroblasts do express the functional CLR and RAMP receptor complexes. So we did the same cyclic AMP, and you can see that they are stimulated by the family to produce cyclic AMP, which suggests they do have the functional receptors. But we're also interested in the role of AM in potentially being able to activate fibroblasts and make them cancer-associated fibroblasts. So what we did was to compare it with a known activator of CAFs, which is TGF-beta, and we used alpha SMA as a marker of activated CAFs. So here you can see that AM on its own is able to activate the CAFs at a similar level with TGF-beta on its own, but then when you put them together, you see the greatest expression of alpha SMA, which suggests potentially there's some kind of a synergy between AM and TGF-beta in activating CAFs. So now the plan is to use our selective small molecule antagonists to determine all these observations, which receptors are actually mediating them. So in order to aid the translation of our AM2 receptor antagonists into clinics, we also investigated the anti-tumor effect on a panel of patient-derived xenografts. So all of these, they express the major genetic subtypes within pancreatic cancer. So we realized that only some of them responded to the treatment, and we, in collaboration with Prof. Andrew Biankin from Glasgow, who determined the genetic background of the ones who responded, we realized that it was the mesenchymal subtype of pancreatic cancer that responded better than the classical epithelial subtype. So crucially, the mesenchymal subtype is actually the one with poorer prognosis, and is actually more treatment resistant. So we had some previous in vitro data actually that kind of adds a bit of more of a dimension to this. So when you knock down REM3, it actually makes the pancreatic cancer cells more susceptible to gemcitabine-induced apoptosis. And gemcitabine is one of the commonly used chemotherapies for pancreatic cancer. So the results on this slide, I hope that I managed to show you that we currently have a patient stratification strategy to select patients who are likely to respond to treatment with the AM2 receptor antagonist, and also a potential treatment combination that we can investigate further. So lastly, this is just a summary on AMs and its very diverse effects within the tumor microenvironment. So the data that we gathered, actually we realized that the effects on the tumor cells were significant, but they were very, very modest compared to the effects on the tumor stroma and the tumor as a whole. So we think, it's because AM mediates all of these different pro-tumoral processes, and this is, shown by our work as well as others as well. But we still don't know which specific AM receptors are mediating these different processes, and our thoughts are constantly evolving about this. So, in addition to the small molecule antagonists that we've developed against the specific CLR ramp receptors, our group's also in developing antibodies against the complex, so not the individual components, in the hope that we can use them as research tools as well. So I hope I've been able to show you the development of the first small molecule AM2 receptor antagonists, and it's been shown to reduce tumor growth in preclinical pancreatic cancer models. And just like to thank my PIs, colleagues, and collaborators who've worked on this project, and also welcome Trust for funding the drug discovery work. And if anybody's interested in any of the small molecules or the antibodies for your own work, please do not hesitate to contact me. And lastly, if you're interested in GPER C6A as a novel testosterone receptor, you can check out my colleague Paris's poster, which is up tomorrow, because it's been shortlisted for a prize. Thank you for listening.
We have time for maybe 1 or 2 questions, if they're short. Any questions?
So maybe I can ask questions?
Go ahead, please.
So thanks. So I was wondering, so it seems that the tumor microenvironment seems to be very important. So what are your thoughts about this, using this antagonist against AM2 and its effect on the tumor microenvironment? And have you looked at the secretome?
Not specifically, but we are thinking about, which avenue to go down. So the first one, we decided to look at CAFs because of the nature of the fibrous tumor, but that's not the only thing we're interested in. I'm specifically interested in the effects on the immune cells as well. So it's been shown that AM can mediate the polarization of macrophages, for example. There's a lot of other little hints in literature with mast cells as well. Infiltrating neutrophils and things like that. So I haven't had time to show the data, but we did do some histology on whether there's a difference in the infiltration of the different immune cells. But unfortunately, when we started this project, we were using immunocompromised models because we just wanted to see if we could, decrease the tumor bulk. But then the more we did it, the more we realized, okay, we need to start using immunocompetent. So I'm very interested in using the KPC, for example, and any other syngeneic immunocompetent to see if we can delve a little bit more on what's happening.
Last question. Yes.
Hey, hi, Heather Giles. Thank you. Very nice, clear presentation. Your original goal with the Wellcome Trust was to develop new therapies for pancreatic cancer. So tell us a little bit about your small molecule. Is it a hit? Is it a lead? Is it anywhere near getting into preclinical development?
Okay, so we have lead compounds basically. So we have a few different series. We're now currently still finessing, doing a lot more preclinical work to kind of choose the best strategy to get into clinical trials. So whilst we initially were developing it for pancreatic cancer, it's we kind of see the potential of this in other cancers as well, because it's not just pancreatic cancer that has the CLR ramp receptor, situation going on. It's a lot of other ones. So right now we are funded by Prostate Cancer UK to look at hormone-resistant prostate cancer. So that's one of the other avenues you might go down. And there's also, as you mentioned, breast cancer is another more fibrotic tumor. So that's another avenue we would go down as well. But right now we have lead compounds that we are still optimizing different things to try and get into clinical trials.
Thank you.
Thank you, Ameera, very much for an interesting talk.
Thank you.
So it's my pleasure now to introduce Martine Smit. from the University of Amsterdam, who's been a real leader in the area of what she's going to talk about, which is viral GPCRs, modulators of cancer, but she's also worked on chemokines and nanobodies and a lot of cutting-edge techniques and ideas, and we're looking forward to your talk, Martine.
Thank you, Paul. Thank you, Paul, for putting this session together. So very exciting. So I will share something with you on unknowns. So I will share with you on the viral GPCRs. And I'll start off with where we stand right now, what we know about viral GPCRs, and I'll share two unpublished studies at the end. So if we look at viruses, especially the herpes viruses, those are DNA viruses containing 1 to 200 genes. They induce lytic infection, but also, most importantly, latent infections. And they have devised strategies to invade the immune system. And it has become across that those herpes viruses do play a role in various diseases, especially in those who are immunocompromised. And also viruses, about 15% of the tumors nowadays, cancers, are associated with viruses. So if we look at the herpes viruses, so we see the CMV virus here on the left, Rosamaria viruses, KHCV, and Epstein-Barr viruses. And those are actually viruses which contain one or more genes encoding for GPCRs. And most remarkably, those GPCRs mimic our human GPCRs, and most importantly, the chemokine receptors. And not surprisingly, they bind a lot of chemokines. Well, knowing that chemokines play a major role in regulating trafficking of immune cells, one can envision that those viral GPCRs may have certain roles. So what are the hallmarks of those viral GPCRs? Well, they're expressed already on the virion of the virus, so they're readily available after infection, and they are newly synthesized after infection. They mimic chemokine receptor homologs and bind a lot of chemokines. And unlike the human chemokines, which show no basal activity and are GI coupled, those display constitutive activity and show promiscuous G protein coupling. So what is the role of those viral GPCRs? Why does a virus carry a gene encoding a viral GPCR? Well, at first it was believed that they act as immune evasion. By binding chemokines, they could escape the immune surveillance. But now it has become clear that they may play a role in cancer as well. So looking at CMV, that's our lab we'll be focusing on. So 50 to 80% of the population is CMV positive. And healthy individuals, CMV infection is asymptomatic. And latently, the CMV virus resides in the progenitor cells. Well, several labs in the US and in Sweden had already detected CMV DNA and proteins in various types of cancers. And we got really excited due to the fact that CMV encodes 4 GPCRs. And we've been studying them, and especially US28 is the most studied. It's a constitutively active receptor. It can be induced by ligands, by chemokines. It's expressed in glioblastoma. And most importantly, recently it has been shown that it plays a role to maintain latency. So in early days, using HEX cells, we transfected it with increasing the concentration of US28, so this viral chemokine receptor, and we saw increases in inositol phosphate production and NF-κB, which is GQ mediated. And whereas the human homologs do not show this increase of basal signaling. But also in infected cells, brain cells here, which are infected with the virus, we see increases of GQ signaling, which is not there when we infect the cells with the delta US28 virus. This is the virus which is not containing the US28 gene. So when we express this viral GPCR into 3T3 cells, fibroblasts, we saw that the US28 expressing cells were growing much faster, showing a foci formation, which is indicative for transformed phenotype. And when injected in nude mice, tumors were formed, which is not the case using the uncoupled G protein mutant. Looking at the signaling pathways activated, it's clear that NF-κB-related genes COX-2 and FHF are highly upregulated. And transgenic mice, in collaboration with the Mount Sinai School of Medicine, Serralia US28 mice targeted to the intestine show neoplasia. And very excitingly, together with Cecilia Söderberg-Nauclear, we had actually detected US28 expression in brain tumor cells, in brain patient material, I should say, especially in the vascular niches. So, glioblastoma is one of the most aggressive types of brain cancer. Survival only 15 months, and current therapy is surgical resection and chemotherapy. But they had already shown in a cohort of patients that CMV proteins were present. And they did some antiviral treatment against CMV, and they saw the prolonged survival. So we asked ourselves, do those CMV-encoded GPCRs enhance cancer progression or not? So we used glioblastoma cells, U251 cells, infected them with the virus, HCMV, and what was apparent that the 3D growth was enormously enhanced. And cells infected with the Delta US28 virus, we did not see this increase of 3D cell growth. And also US28 expressed in U251 cells by themselves already show this 3D increase. And also when we looked at VEGF expression, we saw this massive VEGF increase. Injecting those cells into mice intracranially, we saw that normally those glioblastoma cells show tumor formation after 60 days, while in the presence of US28, we see already after 20 days tumor formation, showing that really US28 is driving this progressing and accelerating this cancer growth. So as said, we are also a nanobody lab, so we identified some nanobodies targeting this viral GPCR to see whether we could actually block its activity. So this is a chemokine binding receptor, so CCL5 binds to this receptor, and we identified nanobodies which could block a CCL5 binding. We made a bivalent, so coupling two together, and we saw this increased affinity for the receptor. And looking regarding its ability to inhibit signaling, it can inhibit the CCL5-induced GQ signaling, but also it's a constitutively active receptor, remember. Those bivalent nanobodies show this inverse agonistic activities. So what about impairing spheroid growth? So when we expose those 3D spheroids expressing US28 to these bivalent nanobodies, we see reduction of 3D spheroid growth, and we see reduction of VEGF production. And also in vivo, we see the mice which are treated with US28 nanobodies injected intraperonally, we saw that indeed impairment of tumor growth. But as you can see, we're not there yet. So what we did, and that's the nice thing about using nanobodies, you can actually engineer them. So what we did, we coupled photosensitizers to them, which one can irradiate, and only those cells which bind to the nanobody will be killed off. So we went back to our US28 expressing cells and our control cell, the parental lines, exposed them to the nanobody with the photosensitizer and irradiated them with the light. And we could see that only the US28 expressing cells were killed off and not the non-expressors. Seen here, those are happily alive, and only the US28 expressing cells which do bind the nanobody are killed off. That was in a 2D setting, but also in a 3D setting, we see indeed that the 3D spheroids, they bind to nanobodies, and when we irradiate them, the nanobodies get activated, ROS is formed, and the cells are killed off. Alternatively, we also devised nanobodies which could bind this receptor intracellularly. And already the Garcia lab had already devised nanobody 7, which actually is binding the active receptor. And we wondered, can we actually find a nanobody which could actually inhibit the constitutively unbound APO conformation? And we did. So we found an intrabody, FUN103, which binds to the intracellular side and recognizes the constitutively active form. So remember, it's a GQ-coupled receptor, and when we genetically express those intrabodies an irrelevant one does not affect GQ signaling, but the FUN103 totally blocks signaling, which is not so surprising because it binds to the site where the G protein is binding to. And nanobody 7, which binds only the activated site, did not inhibit this basal activity. And also looking at 3D growth, expression of FUN103 could actually inhibit this 3D tumor formation. So now we have very nice tools in hand where we could actually look at the contribution of constitutively active activity on one hand and the ligand-induced activity. And we have nanobodies or intrabodies which can discriminate between ligand-induced and constitutive signaling. So, but how does US28 exert the oncomodulatory effects in glioblastoma cells? So what we did, we went back to our glioblastoma cells. We have our parental cells and US28 expressing cells. And what was most remarkably that especially the STAT3 target genes were mostly upregulated, as can be seen over here. And so we already previously had shown that US28 can induce STAT3 activity, and so we used a STAT3 reporter in which US28 shows this increased STAT3 reporter activation. So besides STAT3 is also regulating the expression of sphingosine phosphate-related proteins, such as SK1, sphingosine kinase 1, and S1P1 receptor. And this receptor is a GI-coupled receptor, but it's known to activate JAK2, STAT3, and STAT3 will further enhance expression of SNP1. And as can be seen, cells expressing US28 show this increase of SNP1 receptor and SK1. So it highly drives the expression of these proteins. So we were wondering, so are those US28 expressing cells secreting factors such as SNP1? Because sphingosine is reverted to S1P to activate this receptor. So we took conditioned medium from US28 expressing cells, and we see indeed that SNP1 receptor is activated using this mini GI recruitment. So only conditioned medium from US28 expressing cells were showing this increase of SNP1 receptor activity, which could be blocked by the inhibitor of this receptor. So looking at these pathways, we were wondering, so if we would block, so we have here our US28, which is constitutively active. So could we block at different sites and inhibit this signaling network? So we did on every site, and indeed all of them inhibit STAT3 activity. So meaning that we actually, there's this feed-forward loop which we can actually disturb at these different sites. And indeed, if we block here SK1, so this is so-called sweet spot, if we block it here, we block the expression of SNP1. So as we've already heard earlier lectures this week, well, GPCRs do not only signal on the membrane, but also intracellularly in so-called endosomes. So we were wondering, US28 is constitutively active receptor, and not surprisingly, constitutively internalizing. So we were wondering, is US28 taking along SNP1 intracellularly or not? And it can, US28 actually recruit SK1 to the membrane. So we use this bread setup in which we can look at the translocation of SK1 to the membrane. And indeed, US28 expressing cells show this recruitment of SK1 to the plasma membrane using this KRAS and Venus motif, which is also seen for the histamine receptor, which is GQ coupled. And we looked at endosomal signaling. And there, only US28 is able to recruit SK1 to the endosomes, whereas histamine cannot. So we were wondering, so what about the S1P1 receptor itself? So parental cells show expression of SNP1 at the membrane, nicely at the outer surface. However, when US28 is co-expressed, what we clearly see is that SNP1 receptor is taken along intracellular endosomally. And using bread studies, we checked whether they could form heteromers, and they can. And also looking here, we have the following hypothesis that CMV infection US28 is expressed, forms a heteromer, and takes along the SMP1 intracellular and recruits SK1, and thus can actually activate and initiate cellular signaling. So that's not all, because there are other factors involved in this pathway, and one of them are the phosphatases. So PP2A dephosphorylates AKT and STAT3, but is regulated by the so-called cancerous inhibitor of PP2A, CYP2A. And CYP2A is really highly expressed and especially transformed in aggressive tumor types. And it has been shown to be involved in proliferative signaling. So indeed, in US28 expressing cells, CYP2A is highly elevated, and when treated with the SK1 inhibitor, we see we can ablate or impair this expression. And also by inhibiting and using the FUN103 or expressing FUN103, we can block CYP2A expression and SK1 expression. So next we wanted to know, all those signaling pathways, do they lead indeed to enhanced proliferations? And they do. And upon treatment of SK1 inhibitor, we can really inhibit this US28 enhanced proliferation of these cells. So here, this first part showing that expression of US28 leads to onco-modulation in glioblastoma cells. It's actually driving the feed-forward activation of oncogenic signaling networks. So this SK1, S1P are elevated. More SP1 will activate SMP, and those will actually lead to activations of STAT3. And since STAT3 is regulating expression of SMP1, SK1, we get this positive feed-forward loop. And besides that, also the AKT CYP2A loop is enhancing this signaling moiety. And also by the co-internalization of US28 and SMP1 receptors, we believe that this is leading and shifting to this enhanced proliferation. So looking back at the GPCRs at the plasma membrane and the endosomal signaling and the recycling, what I want to draw your attention to are the so-called degradation patterns and patterns towards MVBs. So what do I mean by that? So in fact, GPCRs do more. They can end up in lysosomals and be degraded, but what they can also do, they can actually be internalized into so-called intraluminal vesicles, into so-called exosomes in these multivascular bodies, and then they can be secreted as so-called exosomes. So this is a new level of complexity. And so what are extracellular vesicles, or EVs? Well, they are actually, EVs is actually the broad name for extracellular vesicles, which can be microvesicles or ectosomes. Those are actually budding off of the membrane, so that's one side. And then you have the exosomes, which are going through this multivascular pathway. And what do they contain? Well, they contain, not surprisingly, proteins which are already expressed in the membrane, but they also take up bioactive cargo, meaning cytosolic proteins, but also mRNA and microRNAs. And the interesting thing is that those EVs, they can propagate signals at short, but also long-term distances. And if we look in a cancer setting, there the biogenesis, so the generation of those EVs is enhanced, its release is enhanced and upregulated in cancer. And looking more carefully at the tumor, we see that by releasing these EVs, they can actually create a so-called tumor microenvironment, and those EVs can communicate with the stromal network. They are believed to play a role in so-called metastatic niches formations, and importantly, they are also believed to play a role in drug resistance, and especially because they have actually devised means to actually lead to immune evasion. So since our receptor US28 is constitutively active, constitutively internalizing, we were wondering, is US28 present in those EVs or not? So we've been looking at them, and so we've been looking at our glioblastoma cells which express US28. seen here in green. And then we use the CD63, which is a marker, it's a tetraspanin, and which is highly enriched in exosomes. And if you can see here, they are co-localized. And using EM, we actually focused and used gold-labeled antibodies to detect US28 in the so-called MVBs, as can be seen over here. And as you can see clearly, US28 is expressed in those so-called intraluminal vesicles or exosomes. So this is still in an overexpressing glioblastoma cell. So what about if we infect those cells with the CMV virus, and we've been infecting them with the virus, and we wanted to see, is US28 indeed also expressed in EVs in CMV-infected cells? And as you can see over here, US28 is highly expressed after CMV infection. And if we co-stain with CD63, the EV marker, we see indeed co-localization. So also in an infected setting, they too are present in those exosomes. So how can we monitor actually this release? So Maarten Bebelman, a PhD student in the lab, he devised a very nice strategy. So we incorporated the protein fluorine, which is pH sensitive, and we inserted into the extracellular loop of US28. So when US28 is in the exosomes or interluminal vesicles within the MVBs, the pH is low, it does not fluoresce, but once it's actually released, it will fluoresce because the pH is higher. And using turf microscopy, one actually can see the release of those exosomes. And if you look carefully, I'm not sure whether you can see it. Oh yeah, here, see? You see some spots lighting up. And those are actually the exosomes which are released in time. So here at different spots, you see this enhanced expression, of enhanced release of these vesicles. But also in CMV-infected cells, so we used our CMV genome and inserted US28 with this fluorine, and we wanted to know whether they would actually, they are expressed indeed, as can be seen here, and whether they could be released. And if we follow and we do the turf microscopy, we see in time that also in CMV-infected cells, these US28 exosomes are released in time. So all well. We see this enhanced release of exosomes containing US28. But what is the functional relevance of it? So as I told you, viral chemokine receptors do bind chemokines. So does US28. It binds CCL5 and fractalkine. So we were wondering, can those exosomes containing US28 still bind chemokines? So we used an N-log tagged US28. And we were wondering, indeed, it can still bind fractalkine. And in the presence of our nanobody, US28 nanobody, we see this inhibition of binding. But most importantly, we wanted to know, can those exosomes bind the chemokine, scavenge the chemokine, and does it have an effect on fractalkine signaling? So we looked at fractalkine signaling here using mini-GI recruitment and exposed them to exosomes without US28, or from cells derived not expressing US28 and cells which were expressing US28. And what we can see here, cells which were exposed to the US28 positive exosomes do show a reduced signaling because US28 is actually binding fractalkine. So it's acting as a scavenger. And if we do that in the presence of our US28 nanobody, we can inhibit the scavenging activity. So here, the key findings are that US28 is present in the MVBs and fuse with the plasma membrane to secrete those exosomes carrying US28. Those US28 positive exosomes still bind human chemokines, and we believe that through their chemokine scavenging, they can actually influence the chemokine receptor signaling. And so they have a so-called decoy function. And so this taking away the chemokines from the microenvironment, they micromanage the TM secretome, and therefore we believe that they may contribute to cancer progression and also to immune evasion. So I hope I convince you that those viral GPCRs, such as US28, do contribute to cancer progression. And I've shown you ways how US28 through the SN1P STAT3 axis and those exosomes, they actually contribute to cancer. And we believe that they might be interesting therapeutic targets to combat virus-associated cancer types, such as glioblastoma. And finally, my last slide, and I must say the most important slide, because those are the people who did the job. So the work on the SMP1 STAT3 has been done by Nick Bergkamp and Jeffrey van Senten. And the work on the EVs done by Maarten Bebelman and Irfan Sitiawan and Katrien Cruden and Michiel Pechtel. And then support from all the other lab members and also Marco doing and looking at the signaling networks in our lab. Thank you.
Thank you, Martine. A very interesting talk on a topic that most people are not very familiar with. It was great. Any questions? Okay, great. Number 2. Peter.
Martine, just amazing. I mean, you got dimers, extracellular vesicles, nanobodies, phenomenal. Just 3 different small, short questions on different topics. So does S1P, you showed that US28 influences internalization of S1P. Does S1P get in the EVs as well?
Good question. We did not check that yet, but I would presume they would, because everything that ends up along, it will get there.
Get further out. And then with that, there's any information in terms of motifs from US28 that tells you how it's getting into EVs and that might be a more common general mechanism?
That's what we're looking at currently,
Okay.
but I can tell you that our thoughts are constitutively active receptors, not surprisingly, that they are actually are skewed towards also this exosomal pathway.
Yeah. And finally, when EVs were first discovered now many years ago, they were thought as biomarkers. That was their great interest, and still today.
Yep.
Have you cross-back referenced with any biobanks or any EVs you could get your hand on to see if you see US28 in sort of patients that are highly metastatic or any correlation on the physiology?
Yeah, very good point. So actually that's what we would like to do, but I must say that mostly the emphasis has been on human proteins. So now we have to look for viral proteins as well. And also same for RNA-seq, it's also been focused on the human genes. So definitely something we would like to pursue. Yeah.
Great, thank you so much, Martin.
Somebody at number 4. I see the 4. I can't see the person.
Hi, Martine. So I guess it's related to the questions that were just asked. I mean, I would expect it's quite difficult to prove a hypothesis in vivo about the relative role of exosomes, or not.
Correct.
Have you thought about how you might do that? Are there specific, ways you can inhibit the release of exosomes and try and manipulate the system to ask a question in that setting?
Yeah. So there are So I'm actually collaborating with the exosome lab. Michiel Pechtel is an important one, and they're looking indeed at ways to sort of inhibit this exosome release and to monitor that. And the people at the Netherlands Cancer Institute, they have used also Cre-Lox mice, in which they did a flip in with tomato green to tomato red, or something like that. And they could actually show that in vivo they could actually see the uptake also of EVs. And it is actually easier to follow apparently in vivo than in a 3D or 2D setting as we are studying.
Yeah. But that shows you the EVs are moving around. It doesn't give you an access to whether your viral receptors in them versus the viral receptors in the cells are doing the business, right?
I agree. So it's really challenging, yes, definitely.
So Martine, I have a really quick final question, which is those, when you describe this photo labeling of the nanobody, right?
Yeah. Mm-hmm.
Can that be used therapeutically?
It could be good. The advantage that you can actually very locally irradiate a tumor. So yes, definitely, that's the way to go, or one of the ways to go. Yeah.
Okay, thank you. Thank you very much, Martine. And our last speaker in this symposium is Rosamaria Lappano, who is from the University of Calabria in Italy. And she is going to talk about G-protein coupled estrogen receptor GPER, also known as GPR30, a mediator of estrogen action in breast cancer.
Good morning, Paul. Good morning, everyone. It is a great honor and pleasure to be here today amongst you. Let me thank the organizers, especially Paul, for giving me the opportunity to make an overview with you on the acknowledged and emerging concepts on the G-protein coupled estrogen receptor in breast cancer. Allow me to say just some words on estrogens to introduce the topic of my presentation. As you know, estrogens are pleiotropic steroid hormones that regulate critical signaling pathways implicated in the control of cell proliferation and differentiation in a variety of tissues, both reproductive and non-reproductive tissues. The biological effects elicited by estrogens are classically mainly mediated by two isoforms of the classical estrogen receptors, namely ER-alpha and beta, which act as ligand-activated transcription factors. Why such a big interest into the estrogen action? Because we know that a prolonged exposure to estrogens is a major risk factor for the progression of breast cancer, which is one of the most frequently diagnosed malignancy worldwide and the leading cause of cancer death in women. As breast cancer expresses elevated levels of the classical estrogen receptor alpha, in approximately 70% of cases, this receptor is considered a favorable prognostic marker because it is associated with an increased survival rate and a minor risk of breast tumor relapse. And this suggests the use of estrogen receptor alpha inhibitors, such as the selective estrogen receptor modulator tamoxifen, in the treatment of breast cancer. Unfortunately, a number of breast tumors does not express ER-alpha, and most of ER-alpha positive breast tumors develop tamoxifen resistance from the onset of the disease or following the treatment. And in this regard, in the last years, it has been established that the multifaceted responses to estrogens can also involve diverse factors, including one member of the GPCR family, named GPER, G-protein coupled estrogen receptor, previously known as GPR30. GPER is unrelated to the classical estrogen receptor, but it acts as an estrogen receptor, as we will see together today. The story of GPER begins in 1997, when an orphan GPCR was cloned and termed GPR30. In 2000, GPR30 was shown to mediate the rapid activation of ERK1/2 signaling cascade in response to estrogens, thus providing the first evidence on its action as a functional estrogen receptor. This discovery was followed by the demonstration of specific estrogen binding by employing both radiolabeled and fluorescent probes, thus leading to the official designation of GPR30 as GPER, G-protein coupled estrogen receptor, and its acceptance by the International Union of Clinical Pharmacologists as a unique estrogen receptor in 2007, thus expanding our perspective of estrogen responsiveness. demonstration of GPER activity as a classical GPCR was provided by estrogen-induced capability to increase the GTP binding to membranes from 293 cells transfected with GPER and membranes from estrogen receptor negative SKBR3 breast cancer cells that endogenously express GPER, as well as by the increase in cyclic AMP content by diverse GPER ligands. Signaling initiated by GPER can occur through diverse pathways and through coupling both G-alpha-S, which leads to the activation of the cyclic AMP PKA signaling, and beta-gamma subunit proteins. Much signaling initiated by GPER implicates the transactivation of the epidermal growth factor receptor. This pathway requires the G-beta-gamma mediated activation of the SRC kinase, leading to the recruitment of the integrin alpha-5 beta-1 protein and the metalloproteinase mediated release of heparin-bound EGF-like growth factor on the plasma membrane, which in turn leads to the transactivation of EGFR and its downstream signaling effectors like the ERK1/2 signaling cascade and the PI3 kinase AKT pathway, which respectively promote proliferative and pro-survival signals. The downstream events of the aforementioned early signaling cascades include extensive genomic regulation. In this regard, we defined through a macroarray analysis the gene signature prompted by estrogenic GPER signaling, which includes a cascade of transcription factors that in turn activate a first and a second tier of genes, including, for instance, the oncogene CFOS and the connective tissue growth factor CTGF, which even today are both considered crucial molecular sensors of GPER action. As a GPCR, the primary sites of the subcellular localization of GPER are unusual, although not unique. In fact, it is mainly localized to intracellular compartments like endoplasmic reticulum and Golgi apparatus, even including the nucleus in a specific cell context. And one of the major concerns for better understanding the roles elicited by GPER is the ability of a variety of estrogen receptor ligands to bind to GPER as well. In fact, despite the differences in cellular localization, mechanisms of action, and overall structures between the classical estrogen receptors and GPER, cross-reactivity of ER ligands toward GPER is prevalent. For instance, estrogens, among which 17-beta estradiol has the highest GPER binding affinity, but also phytoestrogens, xenoestrogens, and most importantly, the clinical used antiestrogens, tamoxifen, raloxifen, and fulvestrant bind to and activate GPER. But selective ligands have greatly aided our understanding of the pathophysiological roles of GPER and in better differentiating the actions mediated selectively by the classical estrogen receptors versus GPER in estrogen-responsive tissues that express both receptors. So many studies have shown that GPER mediates many physiological functions in a variety of systems, including the cardiovascular, bone, reproductive, immune, nervous, and endocrine systems, and it is involved in multiple diseases. The role of GPER has been pointed out in cancer, for which it is emerging as a novel therapeutic target and prognostic indicator. A list of more or less recent observations suggests that GPER manifests biological activities associated with the promotion and progression of diverse tumors, including, in particular, breast cancer. Thus providing a conceivable explanation for both ER-independent and dependent mechanisms of estrogen-induced carcinogenesis. First, it has been shown that GPER expression is associated with clinico-pathological indices that predict disease progression and poor prognosis, such as increased tumor sites, the presence of distant metastasis, and tamoxifen resistance. In this regard, it has recently been found that high GPER levels can lead to a reduced effect of the primary tamoxifen treatment in patients with early breast tumors. These findings agree with our bioinformatics analysis, showing that GPER expression correlates with prometastatic signaling pathways in large cohorts of estrogen receptor negative breast cancer patients, in which we also found that high GPER levels lead to a shorter disease-free interval compared to patients with low GPER levels. Consistent with a protumorigenic role for GPER in breast cancer patients, GPER knockout mouse models of mammary tumorigenesis displayed smaller tumors and, most importantly, exhibited fewer lung metastases compared to wild-type mice. Both in vitro and further in vivo data support that GPER may have a pro-oncogenic function. In fact, in vitro, for instance, GPER activation leads to the stimulation of the above-mentioned signaling pathways involved in cell proliferation, survival, formation of focal adhesions and fibronectin fibrils toward anchorage-independent growth, migration, invasion of breast cancer cells, and tamoxifen resistance. First, in vivo evidence suggests that tamoxifen-resistant xenografts derived from MCF7 ER-positive breast cancer cells regained sensitivity to tamoxifen when exposed to the GPER antagonist, namely G15. In vitro evidence suggests that tamoxifen induces proliferation of tamoxifen-resistant breast cancer cells through a GPER-dependent pathway that involves the PI3 kinase-dependent inhibition of the pro-apoptotic transcription factor FOXO3, and also tamoxifen-induced GPER signaling can also induce migration of breast cancer cells, potentially via a novel pathway that involves the GQ-RO-ROCK-YAPTAZ pathway. So the fact that GPER the fact that selective estrogen receptor modulators like tamoxifen, but also raloxifen, and also the selective estrogen receptor disruptor, fulvestrant, act as ligands of GPER, thus promoting proliferative and, in general, stimulatory signals, suggested that this receptor could contribute to breast recurrence. And a role for GPER has also been suggested in the progression of the triple-negative breast tumor, which is a breast cancer molecular subtype lacking the classical estrogen receptor, the progesterone receptor, and the human epidermal growth factor receptor 2. TNBC accounts approximately 15% of all breast tumors and is considered a major unmet need due to its aggressive features, early recurrence, and poor prognosis. Compared to the other breast cancer subtypes, TNBC has limited therapeutic options for its peculiar molecular landscape and its biological heterogeneity. And in the framework of the transduction network harbored in TNBC cells, which of note are responsive to estrogens, although they lack the classical estrogen receptor, diverse receptors involved in estrogen signaling have been detected, including GPER. In fact, for instance, in vitro, in TNBC cells, GPER activation triggers a variety of protumorigenic signaling pathways like the ERK1/2 signaling cascades toward proliferative events, but also the focal addition kinase signaling to promote migration and invasion in TNBC cells. Of note, we have also found that the hypoxia-induced cooperation between GPER signaling and the hypoxia-inducible factor 1 alpha, along with the interleukin 1 beta signaling, may generate a metastatic gene signature that in turn leads to proliferative and invasive responses in TNBC cells. Consistent with these findings, premenopausal TNBC patients exhibiting high GPER levels show reduced distant metastasis-free survival compared to patients with low GPER levels. Besides, high GPER levels in TNBC cells have been associated with a reduced survival rates, so with a poor prognosis, in patients with lymph node metastasis stage 3 and grade G3 tumors, as well as is associated with protumorigenic signaling pathways in TNBC patients with advanced tumors. Nevertheless, other studies have suggested a tumor suppression action for GPER in TNBC, but also in other breast cancer subtypes. These discrepancies, we think that probably may due to the lack of standardized procedures for GPER immunohistochemical detection and its quantification in tumor biopsy specimens, while the most likely explanation for the differential effects observed in breast cancer cells treated with the GPER agonist named G1 might reside in the high concentrations used and, in some cases, the lack of GPER knockout strategies to test for off-target effects of these GPER agonists. as also mentioned yesterday in its lecture, Professor Gloriam, about the off-target effects of GPCR ligands. So, of course, the role of GPER in the molecular landscape of TNBC remains to be fully explored. And this controversial scenario recently prompted us to generate GPER knockout TNBC cells by employing the CRISPR-Cas9 gene editing system. Whole transcriptome sequencing, complemented with gene ontology enrichment analysis, revealed that GPER knockout leads to a diminished transcription of a variety of genes implicated in cell proliferation. And accordingly, we found that GPER knockout TNBC cells exhibit a lower proliferative rate compared to the normal counterpart in both in vitro and in vivo xenograft models. Moreover, on the basis of our RNA-seq data showing the upregulation of, it doesn't work. It doesn't work. Okay, it functions. Okay. Sorry, our RNA-seq data showed that GPER knockout TNBC cells are characterized by the upregulation of a variety of apoptotic-related genes. And so, on the basis of these findings, in according with these findings, we found that GPER knockout TNBC cells exhibit an increased apoptotic death, and this GPER knockout mediated apoptosis in TNBC cells relies on the mitochondria dependent pathway. Furthermore, we provided novel insights into the role of the gene K signaling in the GPER-mediated GPER knockout-induced cell machinery in TNBC cells. In fact, we demonstrated that the gene K signaling through the c-Jun AP1 pathway promotes the expression and the activation of p53 toward the induction of the pro-apoptotic protein NOXA, which is a main mediator of the p53-dependent apoptosis. In particular, we found that the gene K signaling is required for the phosphorylation of c-Jun, which in turn is recruited to the AP1 site located within the p53 promoter sequence toward the induction of NOXA. And accordingly, we found our bioinformatics analysis of the metabolic cohort of patients, of TNBC patients, showed that high NOXA levels are correlated with a better prognosis in TNBC patients. So in this scenario, we, and in order to dissect the mechanisms by which the gene K signaling may prime the p53 NOXA-dependent apoptosis in GPER knockout TNBC cells, we explored the potential involvement of the cyclic AMP in these effects, on the basis of previous findings showing that cyclic AMP PKA signaling may inhibit the gene K signaling. And so we found that in GPER knockout TNBC cells, we observed reduced cyclic AMP levels content, which in turn stimulates the phosphorylation of gene K and thereafter the c-Jun p53 NOXA dependent pathway in GPER knockout TNBC cells. So overall, these results, which will be published soon, they are accepted, and provide novel mechanistic and biological insights into the role of GPER in TNBC cells, suggesting this receptor in novel therapeutic strategies that could halt TNBC progression. To conclude our overview on GPER in breast cancer, I have to mention that a number of studies suggested that GPER may have a protumorigenic role in breast cancer, also considering that its signaling, its activation promotes the development of reactive cancer stroma. In particular, GPER activation promotes foster cancer-associated fibroblasts, which you know are the most abundant cell type within the breast tumor microenvironment, with a non-protumorigenic role, to produce and secrete a variety of molecules, including pro-inflammatory cytokines, hormones like 17-beta estradiols, and a variety of growth factors, such as the connective tissue growth factor, the vascular endothelial growth factor, among others, that in turn paracrinally promote the proliferation, migration, invasion, and epithelial to mesenchymal transition of adjacent breast cancer cells, as well as tubulogenesis in endothelial cells. And in the framework to provide a potential involvement of GPER into the functional cooperation between breast cancer cells with surrounding stromal cells, with the surrounding microenvironment, we have found that GPER activation may generate a feed-forward loop that can couple interleukin-1 beta induction in breast-derived cancer-associated fibroblasts to interleukin-1 receptor 1 upregulation in breast cancer cells. Moreover, hypoxia, an hypoxic microenvironment has also been shown to increase GPER expression levels, to promote an increased local estrogen production, and also to induce a chronic activation of GPER signaling. And on the other hand, CAFs may be targets of a variety of simulatory molecules released through the GPER signaling by breast cancer cells, as we demonstrated for the inflammatory cytokine interleukin-1 beta, which is released by hypoxic breast cancer cells and released by the hypoxic breast cancer cells through GPER pathway, and in turn promotes the actin myosin contractility and invasion in CAFs. So, to conclude, based on the above findings, novel therapeutic strategies that could halt GPER expression and or its signaling may be useful in the treatment of breast cancer, potentially other types of cancers and other diseases. The idea, a good idea, as it concerns breast cancer, a good idea, at least among those like us who know GPER and its protumorigenic role in breast cancer, is that more comprehensive pharmacological approaches that could target simultaneously both the classical estrogen receptor and GPER might ensure measured therapeutic benefits with respect to the use of ER antagonists alone, which, don't forget, that act as GPER ligands. And in this regard, we discovered a novel compound named MIBE that displays the unique properties to bind to and inhibit both the classical estrogen receptor alpha and GPER in breast cancer cells. And in this regard, also proteolysis targeting chimeras are under evaluation as a strategy for novel anti-cancer therapeutics. As you know, PROTECs are molecules that trigger the ubiquitination of a protein of interest, inducing its degradation via the ubiquitin proteasome system, leading to the downregulation of the expression level of the target and indirectly its function. And so estrogen chimera, first discovered in 2005, have recently, very recently, 2 years ago, recently demonstrated to bind to both ER alpha and GPER and induce their degradation in diverse breast cancer cell lines. So they could provide a useful approach for targeting all estrogen receptors. So let me thank to all the colleagues of my lab that contributed to uncover the role of GPER in particular in breast cancer context, and my special thanks to all of you for your attention.
Grazie.
Grazie a te.
We have time for just a couple questions. We're kind of over late, so please.
Thanks. Nice talk. You didn't mention the potential for this receptor to interact with the RAMP family of proteins, particular RAMP3, which has the potential then to affect its localization. So have you looked at whether or not, particularly given some of the discrepancies in the data, whether or not there's a relationship between the relative expression of RAMP3 and outcomes with respect to this?
Thank you. No, we didn't evaluate this, and no further data in the literature are reported about this. Thank you for your suggestion.
So brief question. So the GPER activity, is it constitutively active, which is driving it, or do you think that really it needs a ligand?
No, it is. I think that in cell context, like breast cancer cell context, but also in cancer-associated fibroblasts, could be probably present a constitutive activity of GPER, but most of the experimental conditions will be performed by adding ligands.
Is there one more question over there? Yeah.
Fantastic talk.
Thank you.
It looked like the pathway that you showed with the knockout cells was upstream of P53 with your diagram.
Sorry, can you repeat?
It looked like your pathway that you showed with the GPER knockout cells was upstream of P53.
P53?
Yes. And so we commonly see that P53 is mutated in some triple-negative breast cancer cells. Have you tested it in P53 mutant triple-negative breast cancer cells?
Mutations, yeah. No, we didn't test, but we know that in the triple-negative breast cancer cells that we used, P53 is mutated. But our data indicate that GPER knockout leads not only to the upregulation of P53 basal levels, but also its phosphorylation.
Thank you.
Thank you.
So I want to thank all of the audience for staying to the bitter end, as it were. Not that this was bitter, but that it went on a little bit long. But I hope everybody gets a feeling that there's a lot going on GPCRs and cancer. So thanks to all the speakers, and thanks to the audience.